We are on site at the Orange County Convention Center in Orlando, Florida at the 2019 ASH annual meeting. We have recapped some of the top news presented each day during the meeting, and later on we'll chat with Dr. John Mascanrenhas on new data in myeloproliferative neoplasms and Dr. Stephen Ansell on the latest updates in non-Hodgkin lymphoma. Welcome to OncLive News Network. I'm Gina Columbus. In multiple myeloma, results of the Griffin trial show that the addition of daratumumab to lenalidomide, bortezomib, and dexamethasone continue to have a significant improvement in response rates and depth of response, as well as stringent complete response and minimal residual disease negativity in patients with transplant-eligible, newly diagnosed disease. The safety profile was also found to be consistent with prior results of this regimen, and investigators noted that the ongoing study has patients continuing on maintenance therapy. Also in multiple myeloma, updated results of the Phase 3 Alcyone trial show that the addition of daratumumab to bortezomib, melphalan, and prednisone, known as VMP, prolonged overall survival compared with VMP alone in patients with transplant-ineligible, newly diagnosed disease. The quadruplet regimen continued to show an improvement in progression-free survival, which was maintained during the subsequent line of therapy. Interim Phase 2 results show that anasinib and azacitidine led to significantly improved complete remission and overall response rates in newly diagnosed patients with acute myeloid leukemia who harbor IDH2 mutations. Additionally, the combination led to significant mutant IDH2 VAF reductions versus azacitidine alone, and it was linked with a tolerable safety profile. The CD123 targeting antibody-drug conjugate IMGN632 demonstrated a manageable safety profile and broad therapeutic window in high-risk patients with relapsed refractory AML and blastic plasmacytoid dendritic cell neoplasm. Due to these data and preclinical findings, the ADC will now be explored as monotherapy in patients with relapsed refractory BPDCN and MRD-positive AML. Combination therapy with navitoclax and ruxolitinib was found to be well tolerated in patients with primary or secondary myelofibrosis who previously received ruxolitinib. The regimen demonstrated clinically meaningful spleen responses, allelic burden reductions, total symptom score improvements, and encouraging improvements in bone marrow fibrosis. Also in myelofibrosis, an analysis showed that select patients on the Jakarta or Jakarta 2 trials achieved similar spleen volume and symptom response rates with fedratinib, whether the JAK2 inhibitor was used in patients who previously received ruxolitinib or were naive to the agent. The findings suggest that fedratinib is a promising new treatment option for patients with myelofibrosis and low platelet counts. In mantle cell lymphoma, long-awaited results of the Phase 2 ZUMA-2 trial showed that treatment with the CAR T-cell therapy KTE-X19 led to significant and durable benefit in patients with relapsed refractory MCL. With at least 1 year of follow-up, results also show that the treatment led to a majority of patients achieving a complete response. It also had a manageable safety profile. Hi, so we are here with Dr. John Mascanrenhas, who's an associate professor in the Department of Medicine, Hematology, and Medical Oncology at the Icahn School of Medicine at Mount Sinai and a member of the Tisch Cancer Institute. Hi, Dr. Mascanrenhas, how are you doing today?
Good, thanks for having me.
Good, thank you so much for being here.
Sure.
So there have been such a nice number of advancements, really, in myeloproliferative neoplasms over the last couple of years. What would you say are some key highlights that have stuck out to you in 2019?
Well, 2019 has been busy with a lot of evaluation of novel therapies, sort of exploiting findings in the lab and bringing them to the clinic quickly. We've seen things from activin ligand traps like luspatercept to address anemia, to BET inhibitors to downregulate the NF-kappa B pathway that seems to be pathologic in patients with myelofibrosis, to antagonizing BCL2, BCL-XL like with navitoclax. So a lot of very interesting things. We just listened to an abstract of anti-CD123 targeted therapy that has been approved in a related myeloid malignancy and now being evaluated in myelofibrosis. So interesting things that you wouldn't even think of, like metformin, for example. In a small study in Brazil, which has some preclinical rationale, and in a very limited number of patients seems to have some sense of clinical activity. And this is a widely available, approved drug. So, novel therapies and then old drugs coming back in novel ways.
Very nice, thank you. So this year we saw the FDA approval of fedratinib in patients with intermediate to or high-risk primary or secondary myelofibrosis. So what are your thoughts on this approval, and can you discuss the analysis of the Jakarta and Jakarta 2 trials?
Sure. So fedratinib is a JAK2 FLT3 inhibitor. It's a multi-kinase inhibitor. It's been in clinical development for a long time, so it's not really that new. The approval is new, which is exciting, as of I think August of this year. So previous to this, we really only had one available approved therapy, Ruxolitinib, for myelofibrosis, intermediate and high-risk myelofibrosis, since 2011, and that's really dominated the space. And it was a great step forward for the treatment of myelofibrosis, particularly as it relates to symptoms in spleen, but is not disease course modifying, and the activity is ultimately limited, and patients ultimately come off the drug at a median time of about 3 years, or probably even less. So we are in need of other therapies, and I think the approval of fedratinib provides us with another JAK inhibitor therapy that's reasonable. The label is broad, so it doesn't stipulate whether it has to be first line or second line. The Jakarta studies, it was Jakarta and then Jakarta 2. I think Jakarta 2 is the one I personally focus on because it was second line to ruxolitinib, and gives rationale for using this drug where the response rate in spleen responses was quite impressive. And the first analysis was like 55% spleen volume responders, and then with some more rigorous interpretation, it was probably closer to 30. Nevertheless, that's an active agent. There are some distinct differences between ruxolitinib and fedratinib. There's more GI toxicity with fedratinib, so one needs to be aware of that and warn the patient and be aggressive with prophylaxis as needed. There's a black box warning of thiamine deficiency. I personally don't think that that's a major issue, but there were 8 cases reported out of like 600 people that were treated with the drug, and ultimately only one case was thought to actually be truly thiamine deficiency. Whether it's really related to the drug or not, I'm not quite clear about, but checking a thiamine level and or repleting patients with oral thiamine is pretty easy to do. So I see the drug right now as a drug that can be used second line very easily for patients who are losing response to ruxolitinib, particularly spleen response or having progressive splenomegaly. And there's an abstract at this ASH sort of exploring the lower platelet dosing cohort, so 50 to 100,000, sort of making the point that there's also that area that the drug can be used in as well.
Definitely. Thank you so much. Now, we've been hearing about some combinations with ruxolitinib for a little while now. So at this year's meeting, we're seeing phase 2 study of navitoclax with ruxolitinib in primary or secondary myelofibrosis. Could you talk a little bit about that trial?
Yeah, so there's a lot of combination studies. Some of them are not necessarily informed by the lab. Some of them really have really sound preclinical rationale. There has always been rationale to combine a BCL2 or BCL-XL inhibitor in myelofibrosis. In fact, Ron Hoffman and his laboratory had shown that many years ago. So the rationale was there. With the approval of venetoclax in AML and a lot of work that's been done in MDS, it sort of was the natural segue that a BCL2, BCL-XL inhibitor would be evaluated, and that's what happened with the navitoclax study. So this is again a relapse refractory myelofibrosis population, and navitoclax was given in hopes to try to downregulate what appears to be upregulation of this anti-apoptotic pathway. The results are interesting. I mean, you definitely have responses, spleen responses and symptom responses. I categorize it kind of in the 30% range. There can be myelosuppression with navitoclax and some other non-hematologic toxicities that one has to understand. But it's clearly an interesting avenue. I think it deserves more exploration, more patients. But I think exemplifies the concept of really, real translational research, and the idea that we really need to be thinking about how to take these drugs and particularly combining them with JAK inhibitors probably earlier on in the disease course rather than waiting for patients to fail JAK inhibitors.
Very good point, and that kind of leads into the next study with the BET inhibitor CPI 0610 in patients with treatment-naive myelofibrosis. So what activity was observed with this regimen, and what questions do you think still need to be answered?
So this is an interesting drug. BET proteins are essentially epigenetic regulators of transcription. they bind to acetylated lysine residues on histone and non-histone proteins. They really have a very diverse function. But what they seem to do is affect transcription of or upregulation of c-Myc and the activity of NF-kappa B, which appears to be upregulated in myelofibrosis and contributes to the pro-inflammatory cytokine storm that we see in these patients that probably mediates symptom burden, progression of disease, fibrosis in the bone marrow. So inhibiting this pathway makes a lot of sense. And we now know that JAK-STAT3 signaling also upregulates and contributes to cytokine production. So preclinical rationale for the combination was provided by Ross Levine and Ria Kleppe in an elegant study that was published some years ago that showed that there was synergy in combining a JAK inhibitor and a BET inhibitor in an MPL-driven murine model in reducing fibrosis, splenomegaly, and cytokines. So this is an example of an informed clinical trial, early phase study, where we took CPI 0610, which is an oral pan-BET inhibitor. It's given in a 3-week cycle, 2 weeks on, 1 week off, and I got to stress it's oral. That becomes very important. And it was 3 cohorts of patients, those that had failed Rux and then got monotherapy, and then those that were failing Rux, they had suboptimal responses, then got combination therapy. We then opened up a cohort of patients who were treatment naive and got the drugs up front. And patients were stratified by whether they were transfusion dependent or independent. What you see basically in a nutshell is the drug has activity. The drug activity is modest, I would say, as monotherapy, but is clearly active as combination therapy in those patients who are having a suboptimal response to ruxolitinib in terms of spleen, symptom improvement. We saw 6 out of the 14 patients who were evaluable who were transfusion dependent become transfusion independent. And we've seen, probably about 40% of patients have at least a grade 1 reduction in bone marrow fibrosis. So we're seeing a whole spectrum of activity in an oral drug that can be added to ruxolitinib with a really good toxicity profile. So very minimal myelosuppression, a little bit of grade 1 to GI toxicity, which is easy to manage. So it gives patients an option, I think, to add something to ruxolitinib to synergize with it. And the cohort 3, which will also be presented at ASH in a poster form, show that you get an 80% spleen volume response rate compared to historics of 30 to 40%. So it's a much more frequent and deeper response than you would get with just single agent ruxolitinib. So those studies will continue to move forward. We will look at more correlatives to look at cytokine expression, et cetera. But I'm excited about that avenue.
Definitely a lot of combinations on the horizon. It's exciting.
Yep.
So what are some other data that you're perhaps authored on that you would really like to spotlight here?
So there's in the non-therapeutic arena, I paired up with a medical student at Sinai by the name of Lucas Rohner. And Lucas was a very ambitious guy with a good vision, and we came up with a project where we wanted to look at whether we could develop a registry of polycythemia vera patients in the U.S., in which we accumulated over 500 patients over a dozen sites. And the question we were asking is, can we go back and look and see, are there real predictors of thrombosis in these patients? Because this has always been unclear and controversial. It's always been assumed that based on a lot of data, good data that's been published in retrospective form, that leukocytosis was a predictor of both thrombosis and progressive disease. And what was shown in this study with Erin Moshier, who's our statistician, who used a very, I think, modern and probably more effective statistical tool called group-based trajectory modeling, that leukocytosis, in fact, did not appear to be associated with thrombotic risk. Progression, yes. Thrombocytosis definitely was not associated with either thrombosis or progression. And I think what the study shows us is that we still don't, to me it confirms that we still don't have a great sense of what are the true contributors to thrombosis in these diseases, which is very frustrating as an MPN physician and frustrating for patients. It really begs the question, what are our goals of therapy and how do we measure them when we treat patients?
That's very interesting. Thank you so much for sharing that.
Yep, thanks, Gina.
So looking back at everything that we've seen in this past year in MPNs, what do you think that we can answer? What do you hope to see within the next year, at the end of 2020?
Well, I mean, my wish list is, which would be any investigator in this field or any physician taking care of these patients. My wish would be a therapy that can be delivered preferably in an oral form, that's convenient, that has a low toxicity profile, that has the ability to have anti-clonal activity, I mean really change the disease fundamentally at its core, to change the course of the disease, to induce a functional remission, something that will be life-extending and gratifying for the physician and the patient.
Very nice. Dr. Mascanrenhas, thank you so much for being here today.
Thanks.
You shared so much great information. Really appreciate it.
Sure, thank you.
We are here with Dr. Stephen Ansell, who is a professor of medicine and chair of the lymphoma group at Mayo Clinic, and he's going to discuss a lot of the non-Hodgkin lymphoma research that's being presented here at ASH this year. Hi, Dr. Ansell, thank you so much for being here today.
My pleasure.
So there is obviously a ton of data in NHL being presented here at ASH. The one I want to talk about first is Mascanrenhas that has some data in poor prognosis patients with NHL, including those who relapse or are resistant to CAR T-cell therapy. So can you discuss this agent and the results that we're seeing this year?
Yeah, this was really exciting data that was presented yesterday. Pretty much it's a bispecific antibody, so in other words, one side of the antibody can bind to CD20, which is on the tumor cell, the other binds to CD3, so it brings the immune cells and it brings the cancer cell into close proximity and gets the whole immune system activated to target the tumor. And it's now being tested in lymphomas. There have been other molecules like that that have been tested in the past with really promising results. These results are particularly promising with a very high response rate, not only in the large cell lymphoma like you mentioned, but also in follicular lymphoma, and they showed some of that data too, and in patients that have had CAR T-cell therapy. So remember, those are those chimeric antigen receptor cells that obviously are targeting the tumor, but sometimes they lose their way and don't really seem to target the tumor like they should. This kind of repurposes them, if you like, and brings them back to where they need to be. And they showed a small subset of patients where, again, patients responded after receiving CAR T-cells and failing. So really promising and encouraging data.
Great, thank you. Now, there's also the Augment Trial, which we saw last year, but this year we saw a subgroup analysis for patients that are older than 70 years old, and looking at that combination with Lenalidomide and Rituximab. So can you talk a little bit about that as well?
Yeah, so I think the encouraging and exciting thing is, could we get to treatments that don't include chemotherapy and are really immunological in nature? So lenalidomide and rituximab are obviously a very good combination. The results are very promising. And as we now have more and more data, it's holding up and showing durable benefit for patients. So this is a very attractive treatment for people that either have failed chemotherapy and then need something else. And as we're kind of looking at it now, it's moving toward frontline, where it's actually been shown to be similarly effective to other chemotherapy-containing regimens.
Great. Now, there's another similar kind of lenalidomide regimen, phase 1/2 trial that we saw with obinutuzumab and lenalidomide. So what's a little bit different about this regimen, and what can we tell about the early data so far?
Yeah, obinutuzumab is really trying to take rituximab's success and make it even better by making it more kind of activating of the immune system. So it switches on macrophages and other what we call phagocytic cells that would attack the tumor cell. And again, obinutuzumab has been shown to be safe, has been shown to be effective therapy, and in combination with lenalidomide, the results now are really looking excellent. And as we're getting more data and it's becoming sort of a longer follow-up, those results are holding up. So I think it's an effective therapy, and I think it's been a promising combination for patients.
Very nice. Now I'm going to move over to CAR T-cell therapy for a little bit. We saw a study looking at earlier steroid use with exicaptogen selalucil in patients with relapsed refractory disease. So could you tell us a little bit about that?
Yeah, I mean, the big problem with CAR T-cells is that you're taking T-cells, you're making them super angry and very mad, getting them in close proximity with the tumor. But when they do that, when the cells are that activated, they have cytokine release syndrome as a problem. We've got around that a little bit with the use of an IL-6 blocking antibody called tocilizumab. But the big problem that we have still struggled with is neurotoxicity. And steroids seem to make a big difference to prevent that. The challenge, however, is steroids could obviously damp down the effects of your CAR T cells. So the challenge is that true? So now, really kind of right after giving the CAR T cells, trials are testing the use of steroids to see whether we can prevent or decrease the amount of neurotoxicity. as it turns out, it looks like it really is having a positive effect on that, and those CAR T cells are so grumpy and so mad that the steroids don't really seem to be slowing them down that much.
I like that you're calling them grumpy. It's a good word.
Oh.
So RGN-1979, that's a bispecific human anti-CD20 and anti-CD3 antibody in relapsed refractory B-cell NHL. So what is the mechanism of action of this agent, and what clinical activity have we seen so far?
Yeah, this is very similar to the antibody we spoke about just a minute ago. Also targets CD20 and CD3. So again, grabs onto the tumor cell, grabs onto T cells, and brings them into close proximity. Similarly, very encouraging results being seen in very refractory patients, and being seen again, being tested, although not presented so much, in CAR T cell failures. So I think these bispecific antibodies are kind of a new wave of therapies that we're going to see really getting the patient's endogenous T cells to do their job, but even sometimes getting chimeric T cells to do their job better.
Wow, very interesting.
Yeah, right.
So now in follicular lymphoma, there's some combination strategies that are being explored as well. So there's lenalidomide and obinutuzumab in the frontline setting and relapse refractory setting, and we also have data that's adding polatuzumab vedotin to that doublet. So what are your thoughts on all these different combination strategies?
You know, I think it's getting harder and harder to keep the alphabet soup all kind of straight in one's head. But I would say that polatuzumab vedotin really looks a very active and useful agent. So targeting a little bit of a different protein, CD79A, at this point, and targeting it by bringing again a payload, a toxin, up close and right sort of into the tumor cell, and delivering a toxic drug that will mess with the ability of the cell to divide. So again, can you have a chemo kind of approach, but very targeted in combination with an immunological approach? That's kind of where the promising effects are. So I think we're kind of excited this time where we have these new agents that really kind of do the delivery of tumor-targeted therapy and immunological activity all together, and we're putting it together in hopefully a regimen that's going to be better for patients.
That's great. Now, in a poster last night, we saw the final results of the Augment trial in indolent NHL. So can you give maybe a little background to Augment, and then what have we learned now with these newer data?
So the Augment trial really is looking at obinutuzumab in combination with bendamustine, and then followed by maintenance therapy, compared to bendamustine alone in patients that had failed rituximab. And the initial studies had shown a substantial difference for the addition of obinutuzumab, and now with longer follow-up, and so important always to have trials that have longer follow-up to show that effects that we see early are maintained. The data continue to look exceedingly good. I guess the challenge is just that not a lot of people will use bendamustine alone, so I think the comparator in that arm is always a little challenging because it doesn't always fit with practice quite as well, but certainly the results are still very good.
Good. Those are some great points. Now, looking back at, obviously there's a ton of data being presented here. Is there any other data that perhaps you're authored on that you would like to spotlight, or any other studies here that you're really excited about?
I think we're learning a lot of things about T-cell lymphomas, so we haven't chatted a lot about T-cell lymphomas, but T-cell lymphomas, I think we learned there's potentially in some of these rare subtypes like NK T-cell lymphomas, maybe changing away from our standard SMILE chemotherapy to a gemcitabine platinum-based combination. There was a randomized study that showed benefit for that, so I think that was kind of encouraging. Old drugs like tipifarnib, which is an inhibitor of kind of the farnesyl transferase mechanism that we kind of had put on the shelf, now revitalized when you've got some biomarkers to look at. So these are things where I think, again, encouraging, interesting new data. And then in Hodgkin lymphoma, a bunch of data, again, just validating the fact that we can move these new drugs into frontline therapy. So the use of brentuximab vedotin, the use of PD-1 blockade, really in early stage patients, frontline patients, has really resulted in excellent results and sustained benefit when we've had longer follow-up.
Wonderful. Thank you so much for touching upon all of that. And Dr. Ansell, we really appreciate you being here today.
Thank you very much.
That's all for today. For more coverage of the 2019 ASH annual meeting, visit www.onclive.com. Thank you for watching OncLive News Network. Signing off from Orlando, Florida, I'm Gina Columbus.